tell one of my favorite stories because I like telling stories. I was very fortunate to be elected to the National Academy of Science in the United States in 2004. And they have perhaps the most boring induction ceremony of any organization in the world. Because what happens in this induction ceremony is 100 people line up, and one at a time, they get on the stage, they shake hands with the president of the academy, they have a picture taken, and they walk across the stage to sign a book. When they're walking across the stage, there's someone with a microphone who reads one sentence about why they got elected. Well, at the time, my daughter was 12 years old, and she and my wife had come to be there at the ceremony. And I knew that she must be sitting there fuming to have to sit through all this boring stuff. So I eventually make my way down to my daughter, and I said to her, you know, I'm really happy you're here to help me celebrate this, but I want to apologize to you because this must have bored you out of your mind. And my 12-year-old daughter looked at me, as she often has looked at me both before and after, as if I am the stupidest person on the planet. And she says to me, Dad, don't you understand about those people? She'd been listening to all those sentences. And I said, what? What do you mean about all these people? And she said a very wonderful and profound thing. She said, Dad, every person on that stage was the first person in the world to see or do what they did. And I think that's one of the real joys we get in science. We get to be the very first person to find something that no one else has found before. It's not a big thing. We're not talking about something to give a prize. It's what we get in the experiments. It's having an idea that works after having hundreds that don't work. It's having a discovery that makes you stop and wonder what's going on. But you have seen it. And I think that's one of the joys we get out of this discovery, is to be able to be sort of unique in that position.
Thank you, Martin, that you said, because in 4 weeks I will be inducted the National Academy of Science in April.
And I will be there to help you celebrate.
Okay, then we will meet each other in maybe in April.
Okay. The next question, still in the open question, what transforms curiosity into discovery that advances knowledge? What transforms curiosity into a discovery that advances knowledge? Who would like to go first? Katalin?
I let Martin to start.
Okay, Martin.
You know, curiosity is fine. We should cultivate curiosity. We should cultivate asking questions. But ultimately, it comes down, it seems to me, to experiments. Actually testing out the ideas. You can have the most I've had many wonderful, truly inspired ideas that turned out to be complete garbage. Not being able to, pass the test of what happens when you do the experiment. So it's really this sort of leapfrogging. You have an idea, you test it out, you change your idea, you test it out, you change it again, and you keep adding to things. So I think the curiosity is what starts it. I think that's wonderful and essential, to be interested, to be committed to what's going on. I think that's really wonderful. But ultimately, you have to test it. You have to put your ideas to the test.
Yeah, you know that all of this curiosity starts with childhood. Every child is curious, want to know, and always ask why, you know. And I was lucky in when I was in school in Hungary, you know, that we went to excursion, and the teacher always didn't give us the answer, just ask us to wonder, you know, about just a leaf, for example. Now it is fall and it is now the leaf is yellow or red, and why it is coming from the color, it was there, it was covered or something, and we didn't get the answer, but we could see also the storks are coming and going, you know, and all of these birds, and then you know how they would find their way back. Of course today kids probably they say that, oh, probably birds had some GPS and they knew where to go back, but you know at that time it was everything was like a magic. How could it be, and why this tree has different kind of leaves than the others, and how the tree would know that this kind of leaves to make, and you know a child is just curious, and if the teachers good to maintain the curiosity, you know, then it will be a curious adult, somebody. So it starts very early on to want to know the answer, and today is easy. You punch in, you know, Google's or what other way, and you are searching for the answer, and you find some answer and you accept or what, but it was growing up in a very rural area, and seeing all of these nature around me was making me always so curious. And my father was butcher, so I have seen him, you know, there was a pig, he opened it, and I was curious to see what is inside made this animal running, you know. Meanwhile, my sister, who later became an economist, she didn't want to see any part of it. But I don't know, I was just kind of different kind of person, yeah. Curiosity, yeah, to maintain it.
I'm glad you brought up the young age curiosity. For to both of you, did you know early on that this is the path you want to take, or is more like no, I don't have this, I better take this.
I was not a child. My father had 6 elementary education and mother had 8 elementary. And we lived in one room with my sister and my parents, and it was an adobe house. We were, you know, we were very poor, but I didn't know everybody around us had a similar thing, and we didn't starve. Some other kids had to go to ask for food for others, but we didn't have to go out, and so it was. I never get a microscope set for Christmas or something like that. You know, it was. And it was just in elementary school, I already, when I was 13, 14 years old, I was third best in the country in biology, in a competition, which was like a 1 week long competition. And later, when I was in high school and I was 16 years old, my biology teacher in this small town, he told me that, Kati, you could be a scientist. I have never seen one, but I imagine, and I start to read what the scientist is doing. They were discovering like vitamins, and this, you know, there were books about, or going in Africa, and I just try to imagine that I will be one of those persons, but I had no clue. And that kind of the teacher inspired me to think about, because my parents would not think about, you know, even finishing high school was something only me and my sister accomplished, because they never been in high school. And that's why, you know, the whole book what I wrote is dedicated to the teachers, all of the teachers, not just mine, but all of the people's teachers that, you know, who help us to navigate in life.
Yeah, but you did say that your curiosity from a young age is already giving you towards that path.
Yes,
Yeah,
exactly.
yeah.
And the teachers helped me,
Yeah,
and
okay. Teachers were
but not in a
support
family
structure.
environment would
Okay,
figure out that I would be a scientist.
understood. And Martin?
So my story is not exactly the same. Um, as far as I can tell, my father annoyed his eighth grade teacher, and she because she was annoyed at him, I don't think he ever went to high school. She suggested he not go to high school. My mother started college, but then couldn't pay the $100 semester tuition fee in the Depression. And so she had to quit and always regretted it all her life. I grew up being interested in science and math. These were things that I enjoyed doing in school. I was not particularly the best at any of these things. But when I went to, I was reminded several years ago by one of my brothers that I did fairly well in school, and there was a big pharmaceutical company in our town, and they gave a fellowship. And I went for an interview, and they said, well, this is a fellowship for you to study some science in college. What science are you going to study? And I had just read the catalog from the college, and it talked about courses in economics and anthropology and all these things that I never had heard of before. And I was excited about all the things I might learn. And so I said to the guy, well, you know, I actually don't know what I want to do. I'm not sure. And he said, you can't tell me you're going to major in a science, which I eventually did. But I said, no. And he said, you're not eligible. Goodbye. And that was the end. And then, I don't want to drag this horror story on anymore. I went to college, I majored, I realized, wow, I better go and work in a lab if I'm going to continue in science. And I went to work in a lab after my junior year in high school. And I had an incredibly bad approach to the whole thing. Somehow in my mind, I had gotten the idea that true scientists worked alone. They never asked for help because they knew what they were doing. So I went to work in a nice lab, although I had to be put in a room that was 50 meters away from everybody else. And I tried to do these experiments. But I never really asked anyone how I was really supposed to do the experiments and what I should be thinking about. So it's not going to come to a surprise to the audience. I failed completely. At the end of the summer, the head of the lab said to me, you know, go home and rest. Come back, try it one more time. If it doesn't work, we're going to have to stop the project. So I rested, was all invigorated. I came back and it failed completely again. And I looked at that failure and I said to myself, I have now proven to myself I should never be a scientist. So my last year of college, I took all these very interesting courses that had nothing to do with science, learned a lot of things, had a good time, and went off after college to get jobs. I had all sorts of jobs, which I'm not going to go into here. And I ended up teaching high school. And when I was teaching high school, I made a discovery. The discovery is, in the summer, students have vacation. Teachers have to find a job. And so I had to find a job, and a friend of mine suggested that I go and work in a lab, that she had a friend, I might hire me. And I went to that lab and had an idea, and I'm not going to go into the idea except to say it was so misguided. It was silly, but it turned out to be right. And I started work. The person who hired me then, 1 week after I was there, said, see ya, I'm going off to Europe to do my own experiments there. So I was sort of left alone, but there were a whole bunch of postdocs and other people. I finally got the courage to ask for help. And I think this is an important thing. You don't do this stuff on your own, or many times you don't do it on your own. And part of the real joys I get in doing science is actually working with other people, where we're all working towards a common goal of trying to find out what a mutant's like, or what a particular situation is, or whatever. So I finally was able to get some help and advice, and I made my own contributions to it. And I have to say, after that, I stayed in science because it was sort of fun to have the experiments work. And I enjoyed it, and I got a little more confidence. My confidence had been completely destroyed by that first summer's work. And I was just very fortunate that I had a second chance. And then after that, when I started my PhD, I had an exceptionally supportive and nice PhD advisor, Bob Perelman, who has remained a friend and was just a wonderful human being. And that helped me gain some confidence, which I sorely needed in this. But so I think you go, people's journeys to becoming scientists and so on, they're not things to be emulated. They're all different. I think we all approach this, everyone's life is an N of one. It's always something unique and different and everything. I was once at a event for high school students here in the United States called the International Science and Engineering Fair. There were a bunch of Nobel laureates, and one of the students asked a panel, or actually one person on the panel said, when you were a kid, you were an Eagle Boy Scout. And I said, yeah. He said, well, how did that help you? And he said, well, I had to do a lot of different things. It was okay. And another one of the panelists said, I was an Eagle Scout too. And I just saw this kid, the gears going in his head saying, oh, I better become an Eagle Scout if I want to get a Nobel Prize. And so I raised my hand and I said, I wasn't, don't worry about it. Just go off and do what you want to do. We all arrive in many different ways, that there is not a direct path.
From both of you, the take that I can make is one, you have to have your innate curiosity, and two, proper guidance.
Have to be motivated, and you have to have, and it doesn't help. I mean, I have to say, listening to Katalin, not getting support from those around you is not fun.
Yes.
On the other hand, I will also say that for GFP, when we first did it, most of the people, although everybody now uses it as a tool, when we first did the work, there were a fair number of people that thought it's just not going to work in our system. They sort of discounted it. They thought, all right, that's all right, but it's not going to be a tool that other people can use. Fortunately, it was. But people, can be real naysayers. You're not going to get the recognition instantaneously if you've done something really interesting. But I'm an optimist, so I think that these things do come.
Yeah, and those naysayers, you do not you listen criticism, but if it is not constructive criticism, just you are saying you are dumb, and don't let other people define you. And if you ask me that what I think it is important for anybody, not even for science, everywhere to survive, I found that mental health, the handling stress. So I was 16 years old when I read the book, How to Handle Stress, and we discussed in high school, and I can handle very well 1930 the word in biology. Prior to that, it was only in physics. And Scheyer, János Scheyer was a Hungarian guy who showed that stress can kill you, not the stress itself, but how you perceive. And you have to learn to handle the stress. The positive stress you need, because that's excitement, expectation, and all of these things. So mental health is a must. You have to be able to handle things. Physical health, I mean, I run a lot. Only one trophy behind me was for running. I run the Philadelphia Marathon, but otherwise, physical health is also very important for everybody. And believing in themselves that they are able to do something, what they are put up as a goal, has to be there, because if they doubt, then don't even start, because probably they will not be able. And all of this life is good to get a goal and go towards, and then learn how all of these difficulties have to be handled. And it is very good, if you move out from your comfort zone, go in another country or go in another group of people to, with sports or other activities, and you will learn more about yourself than like another country. Is very important, because you will discover, just like in the once upon a time tales, that they had just enough food to go to the next village, and then they had to rely on their talent, and they realized that, oh my God, I can do this and that. That's how it happens with scientists and the people, any kind of people. I don't say that everybody had to be a scientist, because probably if they want to be in the spotlight like we are, probably they should be an actress or singers, if they can sing or something, because you don't get the spotlight. I remember when the first time, attention was on me. It was in CNN. I had to answer questions. We are not trained for that as a scientist, and so it was very stressful and surprising for me that I have to say something. And we like to be also to talk to other scientists, to learn from them. But I think that why in the United States we get where we are today, that people are not respecting science and scientists, because we didn't went out to the public. And the knowledge difference between what the average person knows about science and what the scientist knows is so huge and getting bigger and bigger. And if scientists, we don't explain to the public what we are doing, and all of these simplified language, because we have to think about, together with the the reporters, we have to work on it. So we have to help the average person to understand some level the science, because what they learn in school is I mean, when I talked to Drew Weissman, and I was just after finishing the university, getting my PhD, it was like 10 years later, and all of the immunology I learned, it was already out of the window. It was absolutely different understanding of how immunology works. So it is constantly, you have to keep up your knowledge and learn, and so many things, what I found is important. But you have to love what you are doing, and you have to be happy, healthy person. I recently I was on the aeroplane a lot of times, and I can see, the oxygen mask is coming, and next to you sit your child, and of course you try to help. You cannot help. First, you have to have the mask on yourself. So you have to be happy, healthy, before you can help others. So that's what reminds me on that. So that, you have to enjoy what you are doing, get physical, mental health, and believe what you are doing, and things will work out.
Yep. I will add that on. So curiosity, support, and self-perseverance. Keep on going for whatever it is. Okay. I would like to go to a targeted question now. Martin first. Your work with the GFP transformed the way scientists visualize life inside cells. Looking back, can you share a moment when the simple curiosity led you to ask a question that eventually opened the door to this groundbreaking discovery?
So yes, the GFP story. So it's sort of interesting. This is sort of the magician showing them how the magic trick was done. But I have, since I started my postdoc in 1977, working with Sidney Brenner, been interested in the genetics of a set of nerve cells that respond to the sense of touch. And all the time since, we have been identifying genes that are needed for these cells, and by looking for animals that don't respond to touch. And this comes about in two ways. You either affect the developmental program that makes the cells, so the cells aren't there, so the animal doesn't respond, or the cells are there, but what's defective is the response. This led us to go into two directions, which I think is a good way of doing science. Have more than one thing that you're doing at the same time, because they don't always work. So some of the lab was working on genes needed for development of making a particular type of nerve cell. The other part of the lab was looking at how in the world can these cells sense touch? How do they get that mechanical signal to be an electrical signal in the cell? And the mutants we had were doing this. And in about 1989, we were in the process of cloning a bunch of the genes. Now, when you do standard genetics and you clone a gene, you find out what the product is. Almost the first question you want to ask, once you know what you have, is what cell expresses this gene? Is it the nerve cell? Is it an associated cell? Is it something else? Where is this gene active? Now, the standard narrative about biological problems or scientific problems is no one ever thought of that problem before and we came up with a solution. That's complete nonsense. Everybody knew how to answer that question, and we knew how to answer that question, and we did. You could make an antibody against the protein. You could do in situ hybridization against the RNA. You could have a reporter. The one that most people like to use was beta-galactosidase, and looking at XGAL products that were made when the enzyme was made. So you could make transcriptional or translational reporters. All of these would tell you an answer to the question, where is the gene active? The other thing, so I was interested in trying to solve that problem, but we were doing it. At the same time, and I should say something about all those methods are very labor intensive. And the reason they're labor intensive is in order to see the beta-galactosidase activity, in order to see the antibody staining, in order to do the hybridization of the DNA to RNA to look at the in situ hybridization, you have to fix the tissue, and then you have to permeabilize it. And so you get this little snapshot in time, and it's a lot of work. And once you do it, you got to do it again. And then you have to do it again. So, but that's what we were doing, and we were getting the answers. The other thing you need to know in this story is that I've worked since 1977, when I started as a postdoc with Sidney Brenner. I have worked with the nematode Caenorhabditis elegans. C. elegans, as many of us that work on it have bored audiences for years telling about how wonderful this animal is. But one of the things we always say is that this animal is transparent. So I want to know where genes are active. And I work on an animal that for 12 years I'd been telling people it was transparent. I go to a lunchtime seminar, and the invited speaker talks about this protein that I had never heard of before called GFP. And I'm thinking to myself, I want to know where genes are made, they're active, and I work on a transparent animal. This is not deep thought. This is what we were in the middle of. So I'm sitting there, and somebody has just told me that there is a protein that you can see if you shine blue light on it, and green will come out. That was the introduction to his talk. I have no idea what his talk was about. I spent the whole rest of the hour squirming in my seat, thinking about all the experiments we could do if we could put that protein in our animal. I just thought of a whole bunch of experiments. I got really excited about that. So that's the origin of it. After the seminar, I went back to my room and I did what any self-respecting molecular biologist, geneticist would do. I got on the phone. I tried to find out who was studying this incredible protein. Maybe they had cloned it already. They hadn't. I finally found the guy who was in the process of doing that, Douglas Prasher. And we had this wonderful conversation for an hour because we both wanted to do the same experiment. So we collaborated on it. He was cloning the cDNA, and I wanted to put it in first in bacteria and then in worms. So people have sometimes asked me, when did you think it was going to be important? Not important in getting a prize, but something that would be useful to other people. And it was in that first seminar when I was fantasizing about how we could use it if it worked. Because just at that moment, I was really very excited. You can tell from the way I talk, I get excited about a lot of things, but I really got excited about the potential. And that was really the best part. One of the best, well, the best part was it worked. That was the second best part of it. But I loved getting these crazy ideas. And then if you're really fortunate, some of those ideas work out. So that's how the origin story of GFP. You're muted.
You had to have that conference in the guy to spark you off. Okay. All right, that's nice. Katalin, the mRNA technology that you helped to pioneer has changed modern medicine, particularly during the COVID-19 pandemic. At the early stages of your work, what curiosity-driven question kept you motivated when the scientific community was skeptical about this?
I have to say that at the beginning, in the early 90s, there were more publication. Once a year, there was at least one publication out, or somebody was doing something with messenger RNA. They used like influenza vaccine for animals or cancer vaccine in 1995. Those people never published another paper again. And I was, did an early experiment. My colleague, Elliot Barnatan, who was a cardiologist, was interested in urokinase receptors. And because he paid my salary, I said, okay, I can make mRNA coding for this receptor. And I did. And you have to know this protein is only functional when it is post-translational modified, different sugars on it, and the GPI linked to the cell and so on. And what was interesting is that I made the mRNA coding for this receptor, and when we delivered to the cell made functional protein. So the cell was so smart, he knew where to put the sugars, how we were thinking, and how to link, and that early experiment gave me the idea that, okay, we can use in cells, and when the cell is in your hands, like for medical purpose, because I told you that I always wanted to make it useful, was the bone marrow transplantation. So I was reading what the problem is, what should be solved. And there was that older people cannot give bone marrow because the tip of the chromosome getting shorter and shorter. So I thought that, okay, maybe telomerase and the guide RNA. If I deliver to the cells, then I can extend, you know, the lifespan of the cells, and then working on it. So when it was further improved the mRNA and more protein was made, then I can see the animals that like erythropoietin, we could see the effect, we could measure the protein. 30 minutes after delivering the RNA, already you could measure in the blood. And then I was thinking about a disease where very small amount of protein is needed for therapy, and try to make an animal model for that, and so on, so on. I was never wanted to make a vaccine. This was Drew Weissman, who came from NIH, and he wanted to make vaccine, and he was focusing on that, but I made RNA for him also, and we worked together. So it was, I always want to make mRNA that is good for, which has therapeutic protein coding RNA. And so I mentioned my daughter was a rower, and you know, if somebody's a rower knows that all of their, you know, blisters, full of blisters, and I had to puncture the blisters, you know, all the time to my daughter's hand. And I was thinking, oh, if a little collagen mRNA I could inject to these blisters, then it would heal much faster. And I thought that, oh, one day maybe everybody will have a little RNA in their freezer, refrigerator, and when they burn their hands, they just put there, oh yeah, what is this collagen? Oh, 7, okay, I will put a little mRNA, quickly healing. And so this was my thinking was on that way. And so I didn't know immunology deep enough. I learned all from Drew Weissman, and he was the one who wanted to make vaccine. I was improving constantly the RNA structure, new cap analogs were available. I added poly-A tail. I did purification together with Drew Weissman. So we did a lot of improvements. So less and less RNA, we get more and more protein. But in really, I was looking for disease to treat.
Sorry,
And
sorry.
can,
Yeah, sorry. So the take is you had a target to achieve. Yeah. With mRNA technology evolving rapidly, what are the most exciting future possibilities that young scientists should be curious about for the future, for today?
there are hundreds of clinical trials ongoing with messenger RNA right now. More than half of them is infectious disease related, like vaccines for mostly against viruses, but there are also bacteria like TB or other bacteria, acne and causing bacteria, or parasite like malaria. But on the cancer field, you can see headlines that cancer vaccines or mRNA coding for different antibodies are used, or coding for cytokines are used as a cancer treatment. But most recently was excitement about that mRNA was used to generate CAR T cells to treat autoimmune diseases. And also they are using, you know, something which I wanted to supplement certain protein in human which are low level. So there are so many application and possibilities there. It is very simple to make. Delivery is a lot of improvement in delivery in vivo. Now they try to generate CAR T cells in vivo, but you know, injecting the RNA is in a different formulation, which has some target molecule on it, some ligand or something that specific cells are interested. So a lot of science is going on that field. Unfortunately, here in the United States right now is mRNA is something that you shouldn't work on, but most of the science is ongoing in China and other part of the world, and so lot of excitement are there, about how you can use. I have to say that many times I used as a laboratory tools when I had to have some kind of reference material and I want to check whether the antibody works. I just made the mRNA, RT-PCR, and then I made the mRNA and delivered to the cell, and then immediately had the protein made by the cell, so I could check that antibodies are good. Or so very technically I use for many times, and it is very interesting because so instantly translated. So it is like 3-7 amino acids per second. I don't know, as if you have a luciferase 70 kilodalton, I don't know, 15-12 minutes, you can see the protein functioning. So delivered to the cell, or GFP is even, we use EGFP and then it is even shorter, and so you can see very quickly the protein, and even in animals when we injected erythropoietin coding RNA, and 30 minutes later we could measure in the blood. So it is so fast. Yes.
That's quite fast. Yeah, I imagine, Martin, for you it might take a longer time in your work to observe effects.
You mean with regard to GFP?
Yes.
You know, one of the wonderful things about, and I think it was sort of referring to, one of the amazing things that happens after you've made a discovery, gotten a result, is what all the other people in the world do with it. You can think about this with regard to the laser. Charles Townes came up with the maser, and then eventually the laser was made. He wasn't thinking that he was building something that was going to help people in grocery stores get through the lines a little faster, or that you'd be able to hear music or watch movies on these funny little discs that the laser enabled. And he certainly didn't imagine that he was going to be able to, that people were going to use the laser to make a laser tweezers, or to discover gravity waves, or to get low temperature physics. All of these, or all the things that surgeons use lasers for. GFP has been like this. The modified RNAs are also like this. I just heard a talk by Jennifer Lippincott-Schwartz, who has been making probes using the RNAs modified in the same way to really investigate the distribution of organelles within cells, and using these as wonderful new probes. In terms of GFP, I've been completely astonished by all the directions that people have taken it over the years since we introduced using it in 1994. I'm going to give you a few examples to just show the range of things. So almost the very first thing that was done by Roger Chen, was to get a blue fluorescent protein, get another color. So that's the excitation change, a change from giving off green light to giving off blue light in his case. But then he and others also found that you could change not the emission, but the excitation wavelength. And that would allow, because the original GFP was mainly activated by ultraviolet light. Cells don't like ultraviolet light. But it could be changed so it is now only excited by blue light. And people then tried to get other colors, but it wasn't until Mikhail Matz and Sergei Lukyanov in Moscow knew about corals that were fluorescent and got the first red fluorescent protein. This set off a gold rush. People started going into the seas. People started going all over the place looking for other fluorescent organisms, with all sorts of different fluorescent proteins. And there are lots of them, including several mammals that have fluorescent proteins that are expressed in them. In fact, we all have a protein called nitrogen, which looks surprisingly like GFP. And in fact, people have estimated that 4 amino acids need to be changed, and then we'll have GFP in our systems. But there's been so many other things. I'm going to tell you 2 of my favorite applications, neither one of which would I ever have imagined. One is by Jeff Waldo. Jeff Waldo was interested in protein folding, because when you try to make proteins in E. coli and you want to really overexpress them, they often don't fold correctly. And he was trying to figure out a way to be able to mutate the protein so it would grow fast and grow better and fold correctly in E. coli. His solution was fantastic. He made a fusion. At the beginning was the coding sequence for his protein. After that was the coding sequence for GFP. If his protein didn't fold correctly, GFP wouldn't fold correctly. But if he mutated his protein and it folded correctly, the GFP that was attached to it would fold correctly. And he would see that because those colonies would be green. So he could just pick the mutants out that had the correctly folded protein. Thought that was a brilliant idea, having one thing as a monitor for the other. The other thing was Lynn Regan, who was the chair of chemistry at Yale, who did what seems at first like an incredibly silly experiment. She cut GFP in half. Not surprisingly, if you cut the molecule in half, it doesn't work. But then she had this wonderful insight. She attached 1/2 to half of a leucine zipper domain, and attached the other half to another leucine zipper domain that would bring them together. And when they were brought together, the 2 halves came together and a fluorescent protein was there. She discovered a way to look at protein-protein interaction because the readout would be fluorescence. And this split GFP technique has been used, and there's been lots of other tools, lots of other things that I would never have thought of, but I'm really happy that other people have. So these things have sort of a life of their own, and people's own ingenuity and their own problems that they're worrying about lead them into a lot of new directions. And it's sort of fun to be a spectator of that now, I have to say.
With respect to what you've just said, how important is basic curiosity-driven research when especially when the applications are not immediately obvious?
So, as I said at the beginning, I think it's very important to know why you're doing the experiments you're doing. But along the way, you often discover things that you don't expect. These discoveries that Enrico Fermi talked about. And these sometimes have immediate uses and sometimes have a little bit more remote. GFP, I think, is a good example of this. GFP was discovered. We did our work. We published our work in 1994. We did the work in 1992, but GFP was discovered in 1962, 30 years before we started our experiments. And it was done by Osamu Shimomura because he was interested in a fundamental biological problem. How is it that different organisms can actually do a biochemical reaction to produce light? That was the problem he wanted. That doesn't say anything about what that usefulness is, but it's a fundamental problem because The chemistry is interesting. The problem is interesting. And there are so many different organisms that generate light, and they all seem to do it in a different way. So being able to harness the energy of a chemical reaction to generate light is really an incredibly interesting problem. And that's what he was going after. But when he was going after that, that's what he was interested in. He found GFP by accident. Actually, it's two accidents. The story, the Shimamura story is terrific because at first he wanted to know why this jellyfish was producing light. That was the question he wanted to answer. And so when he did that, he spent the whole summer doing experiments, grinding up jellyfish, bribing his kids to get him more jellyfish so he could study them. And when he did, the experiments failed. Nothing ever worked. Spent the whole summer trying to do this. Towards the end of the summer, he's done the experiment one more time, and it's failed. So it's late at night, it's already dark outside, he wants to go home and have dinner. So he takes the prep and he throws it away in the sink. The sink happened to have some seawater, some other stuff in it. He turns off the light and is about to leave the lab when he looks back at the sink. And to his great astonishment, it's glowing. It's producing light. He looks at it and goes, I wonder how that happened. And as he looks at it, he realizes maybe there was something in the seawater. What's in seawater that I've never put in the reaction tube? And he thinks about it and he says, seawater has calcium ions. Sure enough, the next time he tries the experiment, he squirts a solution of calcium ions into the prep, flash of light. The whole reaction needed calcium, something he had never added in any of his preps. And that's what allowed him to purify the light-producing molecule, which was a quaron. The problem with the quaron, though, this is the second mistake, so to speak, is it gave the wrong color. The light-producing reaction produced blue light, not green, but the jellyfish were green. Another unexpected discovery. And he thinks about that for a while, and he realizes, wait a minute, maybe there's another protein that converts the blue light to green light. So he takes a handheld lamp and shines blue light on all the other samples. And sure enough, there's another one. Calcium doesn't do anything for that, but blue light, he gets this brilliant green light coming back. And he, in his 1962 paper, Purifying the Bioluminescent Protein, he has a footnote. The footnote says, by the way, there's this other protein, I'm going to call it the green protein, that absorbs blue light and gives off green. Now, we of course call this GFP. I think he may be the only person that has won a Nobel Prize for a footnote in his paper. But it was just this wonderful discovery. So you might actually ask the question, why did it take 30 years for us to be able to do this, to show that it could be a good marker? What was the difference between 1962 and 1992? And the answer to that is molecular biology. The techniques were not there. Even if you wanted to do that, there was no way of doing it. I actually talked to somebody who had the idea something like 6 or 7 years before we did our experiment. And they said, yeah, we had that idea. We were going to do that. And we looked at it and we decided it was too hard a problem to do. So there's an element of being there at the right time, and having the techniques that allow things to occur and to be able to conduct your experiments. So part of it is just sheer luck of good timing. And the other is thinking about, could this work? Is this something to look into? And I think that's an important thing. I actually also think, I don't want to take up too much time, but I actually think that a very important aspect to all of this is asking questions. And there is so much around us that just washes over us, washes through us, where there are interesting problems. And I'm going to give one example and then I'll shut up for a while. And that is the first lab I ever worked in, the one that started me having feelings that, I mean, first where I actually published something. And I've said this to audiences sort of all over the world. So I'll say it to this audience, but I won't have anyone vote because I usually have people vote on this. I say, the first laboratory I ever got a successful experiment done in, I worked on the transparent part of the body. And I asked the audience, how many of you know what the transparent part of the body is? And most people go, I have no idea what that is. About 5 or 10% of the people, usually it's 5 or less percent of the audience will raise their hand. And then I give them a hint. I say, well, oh, here's a hint. It's right in front of your eyes. Because we're talking about the corneas. But there's an example of a question. It's right there. This is a tissue that is transparent. If it weren't, we wouldn't be able to see. But how many people have actually wondered what makes the cornea transparent? How is it that we can see? What's the organization? Because people have studied this. When I got hired in this lab for the summer, it was an amazing thing to even think that that problem exists. And I felt very silly that my whole life up until that point, I had never thought of that. I think there's lots of these problems that are staring us in the face that we don't even realize are around us. So I'll give the floor over to Katalin.
Yeah, I agree with most of the things, all of the things that you say, that you have to ask questions, you have to wait. I think time is quite an important factor there. And Katalin, what do you think about this?
I mean, I can say to this young audience, probably, I was remembering the late 80s on a way that it was all of a sudden there were RT-PCR. You had a cell line, or somebody gave you a cell line, and you wanted to clone something. If that cell expressed that thing, you just made isolated RNA. There were also RNA isolation. When I was a student, it was not caltropic agent, that it was all of a sudden, we had this simple agent, we could isolate RNA, we could do RT-PCR, we had the sequence gene bank, everything. So it is just, it was so unbelievable, and how helpful the colleagues were, so that when I sent out a letter to send me that clone, the people just put in the bottom on paper and send it to the other person. Everybody wanted to have the other colleagues to advance, and so it was so different. And I might also say that at the beginning you told that I came to the United States in 1985. So for me, the biggest thing was 1985, that I could listen lectures. You have to understand, there were no internet, there were no YouTube. I mean, YouTube, we have 20 years. There were no PubMed. There were no gene bank, nothing. And in 1985. So the only information, the only way you could get, you went to a lecture. And I remember that we drove like 1 hour to listen Bob Gallo telling us something in 1986 that what's going on with the HIV, where is the research is, and we, everybody was sitting in a room because everybody want to know, but otherwise there were no other channels. So it was so different time it was. Today, everybody can listen to any lecture Martin or I gave, and many zillions other scientists, online, and they can listen on YouTube and others, and know that there are so many things that they can choose that maybe they don't know what to choose. But for me, the information was the most important coming here. But in the late 80s, that kind of camaraderie between the scientists, that everybody wanted to have the others and sharing all of these genes and clones and other things, and there were, no signing up and no limitation by the institution, just and it was just unbelievable, this PCR and cloning and other things were so developing, and all of a sudden you had all of these tools. Because I remember when I was student, other students had a project like, killing zillions of animals, harvesting the liver, harvesting some organ, and isolating some kind of protein. That's what, prior to that, scientists did. And all of a sudden, this revolution came. And as Martin explained, that why in the 60s, nothing happened to GFP. And then it had to be, the 90s or, late 80s, when all of this technology became available, and it was small companies was formed. And it was what was also interesting, like publication to make messenger RNA. It was published in 1984, first time sp6 RNA polymerase was used, and in 1985 that T7 RNA polymerase was used to generate RNA. And in the same year, you already, BRL and some Stratagene or other companies, Promega, started to sell. So some discovery you were reading in the publication, and then in the same year, you already commercially available, or, you ask colleagues to send, but, it's so quickly became, like Chomsky cow-tropic agent, or again, all of a sudden everybody started to sell because they realized that it is very difficult to isolate RNA. You had to hot phenol. When I was student, we used a very toxic condition, hot phenol method and other, and all of a sudden you just take out, put on the cell. And so this technological advancement in the late 80s pushed the science and accelerated all of these discoveries, and it was a very, very exciting time. I am glad that I lived that time, and I was, part of it, and I could, benefit all of these methods. I know that cloning was isolating back some insert, from the gel. There were zillion different methods, and everybody had their own, and asked for help, and so it was so excited it was exciting time to be a scientist, and it is always now that all of these technologies available, and it is just so many tools are there. And the question what Martin asked, I can see. I remember my first experiment was when I started in 1978. My PhD program is that from the hospital they get some sample, and they said that because we was a nucleotide RNA laboratory in Hungary, so that these children suffering Leschnian syndrome, and they try to identify that whether this hypoxanthine phosphorylated transferase enzyme is mutated, or whether it was not sufficient. And when the physician told, because I am a PhD, so that told that, you know, these kids are biting off their fingers and their tongue, and I started to, you know, questioning that why we are not eating our own tongue, because when we bite into it, you know, when we are hungry, we would bite into it, of course, you know, we could eat it, but hey, that would be very painful. And why maybe these children don't feel pain. And so that what happened is that there are so many diseases, so many things out there, even today. This is not answered. It is just start to think about that, why certain phenomenon exist and how I can help, or what is the problem. And so it is so many important scientific questions are out there. And then I have to say to all of those students that when they select something to work on, it has to be exciting and important. I feel that way, that you will spend maybe rest of your life studying something, and it needs to be important to feel that importance. But Martin probably think otherwise. But I felt that you know, anything I was doing has to be, and so many enigmatic things, so many disease we don't have a solution, because we do not understand the disease itself, you know, what, yeah, but, you know, people are born with no problem, and then they are 50, and all of a sudden they need a wheelchair, and they, you know, they inherit certain disease. What happened in the first day, at the beginning of the first couple of decades of life? Every day happens something, or all of a sudden they have the threshold, or how? There are so many questions, and that more enigmatic, you know, is more exciting to get answer, and then get a solution, you know, some therapy. And so all of the young students are there, and you know, so many work has to be done, and you know, they are the one who will get the answer, I am sure. I don't know what was your question, but that was my answer.
But I understand that, but what my take is, both of you are saying that the same, both of you are saying the same thing, that there are many things out there, and uh, most importantly is you have to ask the question. You have to ask the question why this is happening, why this is not happening, how this is happening. So the curiosity part comes in that mode, that you have to ask the question, whether who answered it, who answers it, whether the time is right or time is not right. It's a secondary matter, but the question must be asked. That is my take on that.
Yeah.
So I'm going to add one thing to this, if I could.
Yeah, sure.
And is, at least in the United States, I don't know about elsewhere, when adults give advice to students, they say something that I find, and I talk to many students, they find as well, to be the scariest sentence they've ever heard in their life. We say to these people, what we're trying to say is, we're so excited about the opportunities that are ahead of you. Anything, you can do anything. Please, you know, and then they say the scary sentence, follow your passion. I think that's the worst thing to say, because I know if anyone ever said that to me when I was in college, I would have said, as many of the students I've talked to have said, what's my passion? But tell me what I don't want to make a mistake. What if I pick the wrong passion? What if I do the wrong thing? So I think that phrase, follow your passion, is horrible. It's just there to make people unsure about themselves, or at least I would have felt unsure about myself. I think the phrase is something different. It's not follow.
100%, Martin, I agree with you. 100%, I agree that not a passion. Yeah.
But I think the phrase should be passionate. Whatever you're working on. Yes, it's nice to have a problem, but you know, there's so many problems out there that people could do. But whatever the problem is that you're working on, don't do it halfway. go into it fully. There's a lot more enjoyment out of that than holding back. And that's probably also a little bit hard, but I think it's really important to say the most fun, at least as I feel it, comes about by being really interested in whatever problem you happen to be working on and going into it fully. I want to tell you about my favorite experiment from a student. I thought this was a spectacular project. I was at this high school International Science and Engineering Fair, and I was walking around, and there was a student at her booth, and her name was Abigail Freed, and I don't know why I stopped, but I did. And I asked her, "Can you tell me about your work? Tell me what you did." And she started her description of her science in a way that I don't think I will ever hear another person describe their work. Her first sentence was, "Well, the first thing you should know about me, I am a ballerina." Not what you expect from a scientific talk, but that's how she started. And then she said something fascinating. She said, I'm actually at an almost professional level. And when you're dancing as a ballerina at that level, you go through a pair of shoes every performance. And the shoes cost about $125 a piece, for the pair. And so every time you perform, you have to get a new pair of shoes. And the reason for that is the sole in the ballet shoe is made out of cardboard. And it loses its flexibility after a single performance. So what was her project? Her project was to find a better sole for a ballet shoe. So she found a carbon composite material that was too flexible. She looked at 1, 2, 3, 4, 5 different plies of it and found the perfect sole that lasted, in fact, for months. And that was what her project was. She looked around at a project that was interesting to her, and she really did the research to think about how to solve the problem. She's one of my role models. It was absolutely spectacular to see her excited about what she was doing. And I think it was a terrific idea. It doesn't mean you have to go and have all the fancy equipment. It doesn't mean you have to have, working on some really esoteric thing. As I said before, there's questions all over the place. And just recognizing the questions and realizing that you could actually address them is a really important part of what we do.
Martin, what you said about the ballerina, passion and passionate. She was passionate about ballerina, and that drove her passion towards the solution.
Yeah.
Yeah. Okay. To you, still on you, Martin. In your lab, do you or your graduate students drive curiosity, or is it a team effort with questions bounced off each other, questions asked, some sort of thing like that?
It is absolutely that it's really a team effort. One of the things where in my career I have had the most fun has been when students, in one case, and this is continuing because after she graduated, was an undergraduate, didn't have their ego in the game and were just interested, curious, and thoughtful. And when that happens, you get interaction. So let me give a little better description of this. I've had a couple of several people in the lab over the years that have come to me, and they usually come to me and they say, I've got this interesting result. Usually, I listen to the interesting result and say, yeah, I like that. What are you going to do next? And they tell me, and they go off and do it. Every once in a while, they will say what they've done, and I will have a question. And then we have a discussion. Discussion sometimes is called an argument, but we'll call it a discussion right now. And it usually revolves around maybe we need one more control to really know what's going on. And these people all have the same characteristic. As I said, no ego was involved. If they convinced me, that was good. That was the end of that discussion. But if I could convince them, they didn't feel bad that there was something more to do. They simply said, oh yeah, I'll go do that. And they went off and they did the experiment and got whatever result it was. It was really, we were all both working towards a common goal, which was to understand that particular project. And I really enjoy that. I have a technician now who was an undergraduate in the lab, and she stood out because every once in a while she'd come up to me and she'd say, I know this is not part of the project, but I've noticed something. And every time she noticed something, it was just wonderful. There was something great that she had observed. And so I kept saying, yeah, keep telling me these things. I want to know this. And that, I really do think that the very best times are when it really is a collaboration, when it is a common goal that you're working for and not a hierarchy, which I find very difficult. I think that we really, these projects are hard enough that we should be helping each other as much as we can. And I'd like to give a little advertisement here for something that we've been doing in the lab. It actually came from Francis Collins, who was the head of NIH. Many people know about papers that are in bioRxiv, right? They're preprints of papers that haven't gone through peer review, and they're presented. The idea that I got from Francis was to have a journal club in the lab. And we've now done this for about 5 years. In the journal club, everybody in the lab, undergraduates, graduate students, postdocs, me, everybody in the lab, chooses a paper from bioRxiv. In other words, a paper that's has not been published, and we almost always find a paper that's of interest to everybody in the lab. Everybody reads it, and we spend about an hour and a half, one person is in charge, discussing that paper. The person in charge writes down everybody's comments from there's a typo on page 4 to I didn't understand the axes in this graph, or I know of an experiment that they might want to add to the discussion, or many other suggestions. Because these are not from a particular journal, we don't care about whether it's worthwhile going to a journal or not. We're just talking about the science. And once the person, including undergraduates, has gotten all the notes, they write them up and they send them to the authors. The idea being that we're all in this together. We should be sharing our ideas. So if we think, if we're going to spend some time looking at a paper and learning about how to really analyze a paper, whatever suggestions were going to come from that, we're going to send to authors. So we send the authors the comments. We don't publish them or do anything about it. We just make it available to everyone and suggest to the authors that they might start a journal club like this too. What it teaches the undergraduates, a couple of things. One, is that they're learning something, everybody in the journal, learning something while helping other people. And there's something very empowering, I think, when an undergraduate writes a full professor in another lab and says, I was the person in charge of reviewing your paper. Here are our comments. And then a conversation between them comes about. So I advocate this a lot, that, and I don't think it should be done in a department or a large group. I think as a lab, it's a very useful way of getting people to share, which I think is an extremely important aspect of doing science.
That's quite interesting. That's quite interesting. And in the process, the undergraduate might connect with the other person too.
Yes, and no person has ever written us back and said, under no circumstances do I ever want you to read another one of my papers. They've never said that. So, but
Katalin, does this happen in your place too? That you bounce off ideas with each other, or is it personal driven?
I worked by myself when I was doing all of the research. There were no technician, not even students, because I did with my own hands. I was 58 years old. I did all of the experiments.
Okay.
And I had a team when I went to BioNTech, and I um, I like the way to when we did a lot of experiment. It was like a university research lab when we worked there. Then when somebody did an experiment, I never suggested what to do. I always waited that the colleague would come up, who did the experiment. I was asked that, what do you think, what should be the next? But, I had an idea, and sometimes, they said something completely different. But when, they um, make the suggestion what to do next, they will be more enthusiastic to perform, even, we were in a company and is a different setting, then I would tell that do this experiment. So I always found that, working with, I was not a boss there, I was a colleague. Even I was, put to the charge, and I hired the people, and my team, I organized them. But I always was there, because it was in Germany and I had to commute from here. So 5 times a year I came back to my family, but otherwise I was there. And in the laboratory we went, I was always first in the present. And actually I had always breakfast for my team. And then I was the last one to leave because I had no family there. You know, I was living in and working constantly. But when we evaluated the data, and somebody get the result on something we were expecting that day, we will learn. Everybody was hoover over the result and try to see what's going on. But I was probably the last one who said something about it. I let others to, come up with ideas and what should we do next. And so I only enjoyed that time to work in a team. But yeah, I have to say that I read a lot of papers and encouraged others. When I love to read old papers, even in 50s, 60s, 70s, you could feel that more idea, more thought, you were what the authors try to convey or how they understand things, and less data. Today, the papers are 30, 40 pages of data, and you hardly can see what they were, they explain something, but, something that in a big picture to the ideas, you don't see there anymore. So I kind of was amazed at how early on, scientists come up with ideas, and I just could read in their papers, and, they long gone, but, if they would alive, I would go and hug them for how great they were early on. They had this vision. And So I had so much respect for all of the scientists who are not with us anymore. And all of those scientists, you know, every time I get an award, I thanks in the name of all of the scientists, you know, who I read their papers and learned from it, because, you know, this is a team sport. And definitely this is not a 100 meters dash, it is a marathon to be a scientist, and you need a lot of people to work together. And actually my daughter, as I mentioned, was a rower. Actually she just rowing this weekend also in San Diego. But you know, I told her many times, this rowing is similar to what we are doing in science, because the rowers don't see the finish line, they are going backwards. And I told my daughter that I don't even know that when I am pulling my best, whether I am going to the right direction. So, and this is how, and in a rowing, there are never a star, like not it is a team in a, you know, basketball or other team sport, because you had to do everything in a sync. Because if you do much faster, then, you know, the boat would flip over. So everybody had to be at the same speed, the same way to pull that oar. And so it is fun. I like to be a scientist and read about all of these novel discoveries. And, you know, I am excited for all of the young ones who are here listening today, that you know, their life is ahead of them and they can do so many interesting things. And so I hope they listen. A couple of things I also said about, you know, what they need, the mental, physical health, believing in themselves, get out from your comfort zone, and you know, you never know all of those things which is ahead of, you know, your life. I mean, I mentioned that how many times I was terminated, and right now in May I will be honorary doctorate at Johns Hopkins University, which once, you know, 38 years ago, I was had a job offer, but they withdrew because my vindictive boss told them that I am a fugitive and I was subject for deportation. Do you think that I thought at that time, when without no job, no recommendation letter, and subject of deportation, that I would think that, oh, I will come back 38 years later and I will be the honorary doctorate here? No. So there are so many things in life, and in my book I describe also that everything in my life happened that I would you know, I wanted to stay in Hungary. You know, all of these things wouldn't happen to me. So, you know, many things we don't want, but happening, and maybe it is the, you know, our benefit. Just, you know, when Martin described, you know, that things, yeah, so that we are not expecting, but, you know, it's coming, and then we just had to roll up our sleeves and, you know, get over and learn and study and get the best out of it and have fun. Yeah.
Yeah. On that note, both of you have said that get rid of ego. No ego should be involved during the discussion with colleagues, with friends, with students, and all that. So that is an important input from both of you. Okay. Um, we nearing our ending. Just like to ask one or two open questions before we go on to participant questions. Many young researchers today feel pressure to work on safe or fashionable topics. What advice would you give them about following curiosity rather than trends?
You know, that if I consider that what the environment, I myself, I started to work in a lipid laboratory. I was interested in plants, but there were no opening. So I started to study lipids and so on. We put a plasmid in lipid, we covered with lipids the plasmid. And then the next time, you know, they invited me to the RNA team, and that I did my PhD there. So it is, you just go with certain flow and don't, you know, wherever you can get a job or some work, and try to make the best out of it. So, you know, I was not a visionary person. It was just happened that I started with lipids and delivering DNA, and then I went to the RNA lab, and then rest of the history. I just work on those things, and so it is, you know, yeah, I like plants the best, but I never studied them. You know, I still like, I have nice garden and other things, but not studying plant. So that, but my suggestion is that, you know, just whatever is opportunity you have to grab it and make the best out of it. That's what I would say. Martin?
I agree with you on this. You know, this is one of the hardest questions. What should people do? I don't think I have an answer. I don't think I know the right way. I think I rediscover it every time we do something slightly different in the lab. And it's a very hard question. The hardest question, I think, is when do you stop hitting your head against the wall because something isn't working? And it's hard to know when to stop and go in a different direction. And I don't have a good answer for how to do that. People, unfortunately, have to show some sort of progress. Now, if so, I think that really research is a mix. There are some straightforward bread and butter experiments that are important to answer certain questions. And then there are the more chancy things. Oliver Smithies, former Nobel laureate who died several years ago, used to talk about something called a weekend experiment. Weekend experiment is an experiment that you get really excited about, but it's so risky and you're so embarrassed about it that you wait until the weekend when everyone else is away, and that's when you do the experiment. If it works, you crow about it on Monday morning. If it doesn't work, you just ask people how their weekend was. But so I think there is a mix. There is accomplishing things, doing the straightforward things that will answer important questions, but also taking some time to do those experiments that might very well give you novel insights or make those discoveries because they have an unusual answer. I don't think it's doing one or doing the other, but being open to doing more than one thing and chancing it. But you know, people do have to go through university systems and they have to show that they have developed ideas and gotten results. That doesn't mean you do something trivial, but there are things that are more risky and there are things that are a little bit more straightforward. Usually I get my ideas from what past experiments we're doing. You do one thing, it gives you an idea, you go on to the next thing. You read a paper, it gives you some insights into a new direction. But I don't have a good rule about how one goes about doing that.
Yeah, but Martin, you know, then when you are a professor, you have the chance, yeah, I can do that, I can do that. No, the student or the others that, you know, they are looking for a job, they had to do what they get the position for. And they have no choice, and they can love that and that, but, you know, the only opportunity for opening, then, you know, they have to work on that project, what they are hired for.
So I'm going to tell you a horrible thing about me. When I came to Columbia, you know, you got to get everything done to go up for tenure in your 7th year. I did a bunch of experiments. We didn't publish a thing for 6 years. The faculty were really nervous about me. So I understand that, and I understand the pressure that people have, but I also really like trying to finish projects and having things that are not, you know, number of papers and things like that. But I completely agree with you. I think it's really different for different people. And I also know that students, non-tenured faculty members, are under a great deal of pressure. I once thought that it would be a very interesting study to look into all of the Nobel laureates, or maybe a bunch of scientists, and find out how many of them actually made their big discovery before they got tenure or after they got tenure. Because my feeling is that people, once they no longer have the pressure that you're talking about, suddenly get a chance to be a little more expansive, to think, oh, maybe I should try this or try that. And that's a shame, because we should be able to give everybody the chance to gamble on something, to really think about, now this is a very strange thing. I want to go look at this at least some of the time. But it's got to be a balance.
Okay, all right, thanks. That was a good input. We go into, before we end up, we go into the chat box here from Noor Ahmad to Katalin. I've read your book and truly admire your perseverance and passion for science. As a young lecturer and early career researcher, one of our biggest challenges is balancing teaching responsibilities with research productivity. What advice would you give to help us sustain impactful research while still being committed educators? I think the question is
Yeah.
about balance. Yeah.
And then we didn't even talk about family, although Marty mentioned he
Yeah,
has a
you're right.
daughter, I have a daughter,
Yeah.
and so that family and life and the work balance and all of these we didn't even mention anything about. But you know, I found that I was also I never been even tenured track position in my life, and so it was but I was giving lectures for the graduate studies about inflammation, neurological disease and others. So I, is for me it was very helpful to, because when you can explain and teach things, then you have a better understanding, and those are good things. I don't know what kind of lecturing this ask this question is about, but I think that giving lecture is very helpful, connected more with the students, as well as learning on a topic which is not related to maybe directly, but sooner or later you might discover that very much related what you are doing. So I like to give those lectures, but I don't know how basic research or science had to be communicated to the students here. But yeah, everything can be helpful, and so you have to find yourself the right balance, just like with the family, because it is. I am sure that I can tell all of this. I can see a lot of women here. Is that don't try to be the perfect mother and wife and whatever, because you just don't have time if you want to be a scientist. And just you have to find the right partner and together manage. I am married to my husband for 46 years now, and without him I would not achieve and my daughter also would not achieve that many things. So it is very good to have a good family background, and when things are not going well, you know, at least then you have somebody to support you there and help you always, be happy with you, or you know, when you cry, then somebody will be with you.
Yeah, that's a very important aspect of life that nobody discusses. The family aspect that preserves your mental health.
Yeah,
Yeah,
exactly.
yeah. There's an open question. If you become by Adel Majid Kadim, if you become demotivated because the direction of research goes somehow wrong, or you face some problems during the research process, feeling stuck, how do you find motivation again? Does curiosity relate to finding motivation again? Open question, either one.
So I'm going to give a horrible answer. No one is going to like this answer, but I believe it's true. You remember that towards the beginning of this, I said that in my lab, we looked for mutants that were defective in touch sensitivity. And we got some that affected the development and others that affected the function of the cells as sensory neurons. That gave me an idea about how to get over problems. But I'm going to say it in another, even more horrible way. If people look around the labs that they're in, they will see that there will be some people that might be happy, some people that are frustrated. But the head of the lab is usually happy. And the reason the head of the lab is usually happy is at least one person is getting something done. So I'm going to tie these two things together. One way of dealing, and no one wants to hear this, but one way of dealing with the obvious frustrations that are going to come about doing research is to work on more than one thing. If you work on one thing and it's not working, that's a bummer. If you work on more than one thing, for us it was development or mechanosensation, then sometimes one works, sometimes the other. Every once in a while, both of them work, but basically different things work at different times. And while no one really likes the idea of the way to get over being upset about something not working is to do twice as much work, I actually think it's useful to have that extra outlet, to be thinking about something else that you can apply your enthusiasm to while you're mulling over it. I don't know about Katalin, I don't know about other people, but I'll tell you for myself, there are problems I have been thinking about for 30 or 40 years that we haven't really solved. And that those are in the back of my mind, and that every once in a while we get some insight that helps, but we don't get all the answers. But because we're working on multiple things, multiple issues, it allows us a little more flexibility. I'll also refer back to that story about my daughter. When she said these were the first people in the world to do what they did. That realization of being the first person, I think also has within it a kernel of information about why science can be frustrating. If we're trying to be the first person in the world to see or do something, we're not very good at that. And so it's going to fail a lot of times, pushing that boundary of what knowledge we have. When we do push it, when we do gain some aspect of some knowledge about things, it's quite wonderful. But because we're not trying to duplicate things, we're not trying to do old stuff, we're trying to do something new, we are in uncharted territory. Failure is a necessary component of that, because as you push back against all these things, you don't always get it right. You might not have the techniques to be able to do it. You might not, there may be some knowledge that's completely missing. Or your technique might be wrong. Or you're thinking you happen to be unlucky and have chosen the wrong system to study the problem in. Many other possible reasons. But it's trying to overcome that I think is a very important aspect of what we do.
You know, I myself, when I was working, and of course, I was very enthusiastically moved on, because constantly I could feel that the project is working, and the RNA is, you know, whether I optimized, I put new cap on it, poly it, their purification, something, so I could see the progress. It is just other people didn't see, and you know, they could see that I losing my as a scientific standing. Now I was not a faculty, now I am a postdoc, I back like a technician I was working or something, so that it depends on, this is not success if you are looking for that, but you know, this experiment, I could see the progress, and that was important. So if it is, then I start to see the formulation, how to deliver the RNA, and so many aspects I could work on the same one project, making RNA-mediated protein production. So people ask me that, you know, when they were repeating the experiment and they still get the same result, then of course you have to change, because, you know, it's not worse. Yep. That's it.
Okay, yeah, nice. That was a good input from both of you. The question by Park Shen Chung. This one is a bit modern. I was having this discussion with my student yesterday on the topic of AI and education. The discussion led to asking the right question is akin to prompting the right way to ChatGPT. I'm curious about your opinion about how AI is transforming the way we think, do science, and educate. Any thoughts from either one of you? No.
I can tell you about an AI experiment that I'm doing. I don't know what it's like in Malaysia or other places in the world, but in the United States over the last couple of years, every time the news has talked about AI, they have said the problem with AI is students will cheat. Students will have the AI chatbots write their essays. And I thought, this is horrible. Everybody thinks the next generation is the worst type of people in the world, and that's all they're worried about. I don't think that's the problem, because I don't think there's I think that's really thinking about it in a narrow way. If you're worried about students cheating, don't give the students an essay question. Give the essay question to the chatbot and have the chatbot write the essay and give the essay to the students and say, fact check this, because that's the important thing, being able to understand and critically look at things and evaluate that. So I've been saying this for a couple of years. I teach an advanced genetics course. I decided to put this into practice. It has been spectacular. I give the papers that the students are going to read. We have a closed system here, so this is not going out into the public, but Columbia has access to some of these chatbots. And I've given the papers to the chatbots and said, write a 500-word summary of the paper. And they do. And then I give that to the students. And it turns out it makes the students read the papers a lot more carefully, because they're looking for what the mistakes are. And it's been a terrific learning tool. That's not exactly the question that was asked, but we've had a lot of fun with this because they do hallucinate, and they do say these programs do bring up extraneous information, and they don't cite sources. But within this confined way, it turns out to be a wonderful tool to get people to critically think about things. So we've had a great deal of fun doing this in my class over this last semester, and we're going to continue doing it. I think that there are some spectacular things that are happening with machine learning, AI, whatever you want to call it, ways of doing computation and looking things. I think all of us are really in awe of AlphaFold and what has come up in terms of the projections. They're not always right in terms of protein structure, but they have been a considerable way of getting people to think about the proteins that they're interested in, structures, and so on. So I think they're very useful tools. I think they're sort of exciting, but I think, as with everything else, we have to think about what these things mean to our research. I think the biggest problem with AI, you see this all the time here in the United States. Somebody from the government or a lawyer has drawn up some thing, and they've drawn it up trying to get their work done really fast, and they've drawn it up using a chatbot that made up things. And then they're called on it and they look really foolish because they weren't careful themselves. I think we really now have to start training people not to not use these things, but to critically use these things. And I think that sort of approach can help. Already, it is improving the grammar of lots of student composition. I have no problems with having these things talk about how to better phrase something. But underlying all of it is understanding. Now, I'll say one other thing. A dear friend of mine has a teenage son, and her teenage son wrote an essay for school that my friend watched him write. He turns it in, and a couple days later, the teacher calls my friend in and says that the teacher was worried that the students were going to cheat. And so she asked ChatGPT, "Did you write this essay?" And she told my friend, "Your son's essay was written by ChatGPT," which she knew was not the case. The problem here is not ChatGPT. It's not the student. It was the teacher believing that they were getting truth and an honest answer. It again goes back to the critical thinking, I think is really important. But tools are there, we do essays. I'm sure Katalin will remember when, as I do, when all of a sudden all these wonderful DNA and RNA kits came available. And students suddenly didn't know how to do the experiments, but they knew how to use the kits. And you'd ask them, well, what's the basis? Or how do you control for this? Or what should you be watching out for? And it took people a while to realize how they had to be critical while using a kit to make DNA or make a prep of various types. That's the same thing I think we have right now with AI. It has the potential of being a terrific tool, but it's only when we can evaluate and think about what the output is. Anyway, that's my take on this.
Okay, all right. One last one for Katalin by Quack. As a former member of the Women in Physics IUPAP WG5, what more should we do to promote women scientists?
Yeah, so if I don't know, is somebody from the government is listening here? I don't know, there's any person here. So that for me, it was wonderful that when I had my daughter, we had affordable, high quality, affordable childcare in Hungary. So at a very nominal fee, 5 o'clock in the morning, I could take my daughter there, and professionals, nurses were present when, she was like a couple of months old, and every day a pediatrician visited the facility. So, that I could send a message. They gave all of the vaccination to them. I just had to sign that I understand that she get. So it was such a thing would help not just scientist women, all of the women would be helped if there would be, government supported high quality, affordable child care would be there. And yes, of course, now that all of these women who is still listening, you have to make sure that you find the right partner, because your partner could help you a lot, because if you have a family, child, and, you have to make sure that you are together you raise. And then I know that when the parents, elderly parents are sick, or everybody is looking at the woman, the woman should be there and assist and help, but it is should be, it is more cultural that so the husband should be participate all of these activities and help. That's and of course the government could help also with and then it's when I would also locally give more money, like in an institution could, if they have some extra money to give scientists, that when they know locally that, okay, there is a big gap in the publication list because she delivered 2 or 3 child and then couldn't be in doing experiments that help them to get back. I know I worked in, as I mentioned, in Germany, and then there were paternity leave also, that so the father could leave a job for 6 months, the mother could leave, because, if a mother would be a scientist and would go away for 2 years, it is impossible to get back. It is so difficult, so hard, and and I also have to consider that some person want to have a family, and then just raise the family. The other want to be scientists, but you can be both, but then you have to make sure that your partner and also have, and you do it together. My daughter turned out also okay, and probably Martin's daughter is also fine, and so it is. It can be done, but definitely, that when I am as a woman, they more likely ask me. So how could you do that? That your daughter and when a male scientist are there, nobody asks, oh, you have 5 children, how can manage? Yeah, probably the wife is at home, managing, and they never question a male scientist with children that how they would manage. Do they ask ever, Martin, that how you managed your daughter?
What I get asked often is sort of the balance of work and life.
Mm.
And if I could add 2 things to your thing, I'm going to tell my parents. My parents, when they got married, after they had their honeymoon, they came home. Both of them worked. My mother worked, they actually worked together for many years in the same company, but my mother worked, my father worked. They came home, my mother cooked dinner, and they had dinner, and my father said, "This was wonderful, thank you, this is a wonderful meal." And he got up and he went into the living room. We're talking 1945 here. Goes in and walks into the living room, sits down and starts reading the evening paper. My mother came up to him and said, "You know, We both work. I don't mind cooking, but I think you should do the dishes. My father looked up at her and said, "You're absolutely right." And for the next 50 years, that's what they did. So that's my training in this, is that it is a shared experience. And that's what you were saying before, because they both did things. Actually, my mother ran the company. My dad was a salesman in the company. So that is, one of the aspects. But there's another thing that I've, over the last couple of years, become very interested in. And this is the work that I know of, started with a man named Enrique de la Cruz at Yale University. He wanted to have, he's the chair of the molecular biology and biochemistry department, and he wanted to have a more equitable way of hiring faculty. And so what he did is sort of taking a cue from what orchestras are doing, where orchestras now don't look at who the people are that are auditioning from them. They have a curtain. You can't see the person. You don't know who the person is behind the curtain. You can just hear them performing. You can just hear their audition, and you judge by the music. Well, that's what he did for his department. They sent out letters to people saying, "We'd like as many people as possible to apply, but apply in a way that doesn't tell us anything about where you got your degree, where you've published your papers, or anything. All we want is a statement about what science you've done and what you want to do, but don't refer to the papers or who you've worked with or where you've worked. We just want to hear about the science." And they had a separate essay, I think, about teaching, of what they wanted to do. They then looked at these anonymous statements and decided on who sounded the best in among all the applicants. And they narrowed it down to a group of, I don't know how many people. And those are the ones that they had get letters of recommendation, and then they found out the publication. They invited them to give talks and all the usual things that's happened. But the first step was the anonymous evaluation. And what they found, to their astonishment, was that for the first time in the history of the department, half of the people that were in that final group were women scientists, and 17% were underrepresented minorities. In other words, when they went to look at the science and not were biased by did they go to this university or that university, they picked the people they thought were the best. And it turned out in that case, and I think our Department of Astronomy here at Columbia has done the same thing, that they wound up asking an amazingly more diverse group than they had ever done before. So I don't know that this will happen every time, but I think we don't even know the subtle cues and problems and everything that come up. So I'm really quite curious about these sort of anonymous first steps in hiring people. And I hope that it brings a more diverse group of people into science and shows the recognition that their science deserves.
Thanks, Martin. That's a nice one. That's a nice closing about this one. There are two more that I won't read. I'm sorry, Nordin and Mohit, otherwise we are a bit late. We thank thank you both our panelists for very much for the insights and the lively dialogue on the curiosity and discoveries. Before we bid you farewell, we'd like to have a group photo as is usual. Please, listeners, all of you, could you please turn on your cameras? Yeah, quite nice to have you back. Thanks. Please, Dr. Pinko is here too. Okay. All right, please, guys, please turn on your cameras, 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 cameras. Cameras, cameras, cameras. Bit more. Cameras. Shah, whenever you're ready, please just give us a cue.
Okay, all please open camera.
All camera on. Done. Okay. Nice to have you all here. Thanks for
Done.
joining us. Okay, all right. Thank you, thank you, thank you very much. Thank you, Martin. Thank you, Katalin. Good night to both of you. I'm sorry we extended a bit more than we were supposed to be. Thank you very much. Nazri has left. He had something to do, but he will get in touch with you with the certificates. Thank you, Martin. Thank you, Katalin, for joining us.
It was a pleasure.
Thank you. Bye.
Take care, everybody.
Thank you very much for the beautiful insights. And for the rest of you, the attendance link has been given on the chat box. Please click on it and get your attendance, please. Thank you.